All right, this video is going to get into one of those things that's not quite a sense of its own in the technical meaning, because it doesn't have its own receptor system, but rather it's a perceptual experience built out of input from our other sensory systems and what their detectors pick up. So what is flavor? We can define flavor as a multisensory perceptual experience combining primarily taste and smell, but as we'll see, also other sensations, including vision, audition, mechanoception, thermoreception, etc. So when we're eating food and saying, this tastes amazing, what we really are saying is this flavor is amazing, since taste only boils down to some combination of sweet, salty, sour, bitter, and umami. Whereas the vast variety of flavor experiences we can have absolutely dwarfs the simple combination we get from those 5 basic taste qualities. So this is one of those places where our colloquial everyday use of language is a little different from the scientific use of those terms. Part of what makes ice cream so damn delicious is not just that it's activating the right proportion of sweetness and saltiness receptors on our tongue, but also the odorants that are coming off of it, and the texture and temperature and mouthfeel of it too. If you have a cold or you just plug your nose, ice cream isn't as enjoyable. Not because it tastes any different, it still turns on the same taste receptors of sweetness and saltiness and stuff, but because the flavor is different. Now, the primary components making up our flavor experience, they are, as you might expect, taste and smell. The taste one's obvious, it's our mouth, right? But why is smell involved in the experience we have when there's food in our mouth? Well, as I mentioned in a previous video, our nasal cavity is actually connected to our oral cavity, our mouth cavity. Air travels between our mouth and nose. In other words, sort of way in the back there, we've got a connection between the two. So when we're chewing food in our mouth, a lot of odorants, a lot of molecules are being released and going up that back passage to hit the nasal cavity and land on those olfactory receptor neurons, the ORNs of the nose. We call this process retronasal olfaction. Just means back nasal, right? Back nose olfaction. Normal olfaction would be air coming in the nostrils when you breathe in at the front of your face, whereas retronasal olfaction is the other route to get air into your nasal passage. Turns out this is actually very important for our experience of flavor during eating. Just for example, some studies have looked into this, how we perceive the flavor of something when we do have retronasal olfaction. So when our nostrils are open and air is flowing kind of freely between those cavities, versus when we clamp our nostrils shut and so air doesn't flow freely back in that retronasal way to get into the nose. So for example, flavor perception for the exact same food or compound is totally different when the nostrils are clamped shut. On the left here, we've got one chemical. It's sodium oleate. What most people, if you just put it in their mouth, they would describe it as a soapy flavor, like soap when it's in their mouth. The X's here are just a bunch of participants in this study. They're just what descriptor a given participant in the experiment used to describe the flavor when we put sodium oleate in their mouth. So in the column labeled open here, that's when they describe this chemical normally, with their nose open. But the column labeled clamped is when their nose is clamped shut, so air doesn't move as freely in the nasal cavity. We're getting less ability to pull air up in the back retronasally, and basically by clamping their nostrils, you're largely removing the smell component of flavor, right? Then it's just pure taste. And when you do that and you ask people what's the flavor of this stuff when you put sodium oleate in there, the soapy flavor, right? It's no longer described as soapy by most of them. Instead, most of them will say it's tasteless, or what they mean is flavorless, right? Same thing for the next chemical here. So ferrous sulfate, usually put that in someone's mouth, they will be like, oh God, that has a metallic flavor. But if you clamp their nose, remove the retronasal olfaction because air isn't passed up from the oral cavity to the nasal cavity, suddenly they say it's tasteless, or most of them. They're more likely to say it's tasteless, meaning, again, flavorless. They're not experiencing anything very strong. Now, there are some exceptions, like MSG, monosodium glutamate. It gets described pretty similarly whether there's retronasal olfaction or not, but that's probably because the flavor of MSG is pretty largely coming from taste rather than smell. It's coming from taste, like the basic taste qualities like umami and saltiness, that saltiness being like the sodium in monosodium glutamate, and not so much from smell. So that one doesn't have as much of an effect. But for most things, the flavor is coming from a combo of taste and smell, as we can see by the difference when we do have the smell component there via retronasal olfaction or don't. Now, I want to mention a pretty interesting perceptual illusion that happens with flavor. Now, you might remember earlier in the course, in previous videos, we learned about one sense kind of capturing another sense. Like when two senses are giving us input that disagrees about what's happening or where it's happening, one of those senses tends to win, and that's what we end up perceiving, right? So, for example, the rubber hand illusion was an example of this. In the rubber hand illusion, we feel the touch in hand-centered, body-centered coordinates, and then proprioception is telling us the touch is happening far off to the side, where our real hand truly is hidden off to the side there. That's what proprioception says, as my hand is way over here, which is true. But because of the fake rubber hand that we can see being touched in the same way, vision is telling us that, oh, that touch is happening here where the rubber hand is. And in that case, in the rubber hand illusion, vision wins. Vision captures proprioception, we say. Vision takes over, and we have an illusion of feeling like our hand is somewhere where it's not. We feel like our hand is closer to where we're seeing the touch happen. That's visual capture. Another example of visual capture, there's something called the ventriloquist illusion. This happens in like a movie theater or even at home when you're watching your TV. But an obvious one would be if you're at a big movie theater, you're watching a giant screen in front of you. When the actor on the screen is talking, the sound of their speech, the auditory sound of their speaking, is coming from speakers that are off to the side in the theater setup, right? The sound is not coming directly from where their lips are on the screen. The actual sound is coming from the sides. But visually, the speech is happening where their lips are at on the screen, right? So two different senses are localizing the speech as coming from two different parts of space, two different places. One of our senses says, oh yeah, the sound, the talking is coming from over here, and one says it's coming from over there. And in that case, when you're in the theater watching the movie, you experience the speech as coming from the screen, where the lips are moving, even though that's not where the sound is actually coming from. So your eyes are overriding your ears in the ventriloquist illusion. In other words, vision is capturing audition in that case. And in other cases, a different sense might win. When two senses kind of disagree, or if we set things up in a lab so that two senses disagree, we'll often find others might capture vision as well. So anyway, that's the idea of capture. Well, with flavor, we've actually got one of those same situations happening. The taste signals of our food are coming in at the tongue, right? But the smell signals are coming in at our nose. And those two things combine to perceive flavor, right? We perceive flavor as a combination of taste and smell. But where then do we perceive that flavor? Do we perceive our flavor in the mouth or in the nose? And in this case, if we look at the data, we seem to generally experience flavor as occurring in the mouth. So this is actually called oral capture. Sort of like the mouth is capturing it. What we really mean is taste is capturing smell, in this case, in terms of just localizing where the flavor experience happens. Think of it like this. The taste experience is like happening here in the mouth. The tactile sense, and yes, we have touch receptors on our tongue. The tactile sense says the experience is happening here. The food is here in the mouth. But smell, it says the experience is happening over here in the nose where the olfactory receptor neurons are going off. So the brain has to solve this dilemma, this disagreement between the senses so that we can have a unified perceptual experience, not some fuzzy, vague, kind of spatially confused experience. And in this case, it basically says, okay, let's perceive the location of this multisensory flavor experience. Let's perceive it here in the mouth. So the mouth is usually flavor town. That said, researchers like to mess with this stuff, right? To understand the mechanisms of how the brain works. So experiments have done things like vary the time delay between when something hits the odor part versus the taste part, when it hits the nose versus when it hits the taste receptors. So we're kind of timing it out. So do the taste receptor cells send their signal first, or do the olfactory receptor neurons send their signal first? And what do we find? When we present both of them simultaneously, when both neurons fire at the same time, people experience the flavor as occurring at the back of the mouth, sort of spatially towards the back of the oral cavity. But if we present the odor first and then the taste cells go off, we actually experience the flavor as happening in the nose. And if we present the taste first, if it comes much earlier than the smell, we experience it as spatially located closer to the tip of the tongue. That's where the flavor is happening instead of at the back of the mouth. So in normal circumstances, right, there's a consistent and predictable pattern of timing between when taste cells fire and when the ORNs in our nose fire. But if we mess with that, we see that the flavor experience changes too. Now, before we move on from this idea of retronasal olfaction, I wanted to give you an idea of a fun and kind of easy flavor experiment you can try at home. The task is really simple, jelly bean flavor identification. So to do this, you'll want to buy some jelly beans that have different flavors. I like to get at least a little handful of some very common and clearly different flavors, like cherry, lime, lemon, vanilla, and pina colada, for example, something like that. But you could use any variety pack you want, as long as the flavors you're using are ones you would normally be familiar with, that you would definitely recognize the flavor of normally. Now, the best way to do this experimentally is to partner up with someone to test you. Do it with like a friend or family member or something, but you can easily do this solo if you prefer. You'll still get the same idea out of it. At any rate, as the participant, in any case, just as a participant, you want to keep your eyes closed for this. On every trial, you want your eyes to be closed during the trial so that you can't identify visually which jelly bean is going in your mouth. You want this to be a pure flavor kind of thing. Now, if you've got a partner, then they're the experimenter, right? They're going to randomly pick one of the jelly beans and maybe put it in your hand while your eyes are closed. Because again, you're blindfolded, so you're doing this by touch. Or if you're solo doing this by yourself, you can just grab one out of like a mixed pile, but do it randomly so you don't know which one you grabbed. And then for each jelly bean, what you're going to do is before you put it in your mouth, clamp your nostrils shut. Clamp your nostrils shut. You're going to try not to breathe in and out too much during this. So even with your mouth, you don't want to breathe a whole bunch because you don't want to get too much of that retronasal olfaction. We're trying to block that experimentally. So for each jelly bean, you clamp your nostrils shut, then put it in your mouth and just chew it for a few moments. Again, with your eyes closed, but also your nose closed, and try to identify the flavor. Then what you could do is, after you've got your best guess or whatever, then unclamp your nose. Now breathe. Unclamp your nose, breathe freely, open your nostrils while you're still chewing, and try to identify that same flavor, right? The same one that's still in your mouth. Usually at this point, it'll be super obvious what flavor you're doing. So you could, if you want to kind of make an experiment out of it, you could mark your initial nose pinch guess, right, as correct or incorrect. Or if you're doing it with a partner, they can write it down. But you just kind of repeat this for a few different flavors. Maybe repeat it for the same flavors a few times randomly mixed together so you get better data over time, right? That way the participant can't guess what's coming. You don't know what you're going to do next or what's left. You're just doing a totally random mix of a whole bunch of these. And just see how accurate you are with your nostrils pinched shut versus when it's open and you have that retronasal olfaction. It should be pretty clear that the smell component is a huge part of our flavor experience. Like, that's the thing people usually call taste in the colloquial use of the word. It's super different without smell. In fact, we know that the taste experience doesn't change here when you pinch your nose. What you'll probably notice in the nose pinched condition is that all of the jelly beans just taste sweet, sugary. That's because your taste receptor cells, remember, they only pick up the 5 basic taste qualities. Your taste receptor cells, they can pick up that basic taste quality of sweetness without any smell needed, but that means all of the jelly beans are just going to taste like sugar, right? We still get the taste, so we get sweetness, but man, that smell adds a whole hugely important other dimension to our flavor experience. And that's what makes a bag of jelly beans not all taste the same normally, or I should say, not all have the same flavor normally. Indeed, all modern food products use this knowledge, and we can demonstrate this stuff really easily in a controlled laboratory setting. Just for some classic examples, the sweetness of sucrose, meaning like how sweet like how sweet you find a specific amount of sugar, whatever the amount of sugar is, maybe in a sugar water or something like that. We set it to a certain level and just ask people, how sweet do you find this? That sweetness doesn't just come from the sugar itself, but for the exact same amount of sugar in the water, we can get different ratings of sweetness just by also adding in some smells like strawberry or vanilla. And that effect actually goes away if the nostrils are blocked. So we know it's not due to an actual chemical interaction. Also, we know it's not due to just a sort of sensory summation, just adding them up, because it's specific to like taste odor qualities. Like a strawberry smell enhances sucrose's sweetness, but doesn't enhance its saltiness. Whereas peanut butter odor doesn't have that effect. So smell doesn't just like add into our flavor experience on top of taste. It can actually alter the way we experience the exact same taste receptor activity. The same taste qualities get experienced differently when in the presence of complementary smells. And indeed, a loss of smell, which is called anosmia, lack of smell, drastically affects our flavor perception. And functionally, what ends up happening is it lowers appetite. We end up with a lot of weight loss and malnutrition, which is very similar to what we saw for ageusia, the loss of taste sensation. So that's because losing either of those senses is going to have a huge impact on our flavor perception. We need both to have the full flavor experience. Okay, now, considering that the pathways for both taste and smell come into the brain at different places, how in the world do we get this interaction of the two within the brain? Well, let's go back. Let's go back to our mapping of brain areas that we saw with olfaction, with smell. Info comes into the olfactory bulb, right? That's the part at the kind of long yellow pieces here at the bottom of the frontal lobe, right above the nose. So info comes into the olfactory bulb, and while it goes to a variety of places, we saw the most important next step is the piriform cortex, where we recognize the overall pattern of a smell, just across a whole population of neurons firing together as a consistent set. And that lays down, oh, it's this smell object, right? From there, you can see it does connect to the amygdala and indeed other limbic areas too. But one of the other major areas it goes to next is called the orbitofrontal cortex, what I've highlighted in red here. This is the area in the brain where taste signals and smell signals both really combine together to process flavor effects, as opposed to just processing their own signals kind of separately and independently. So the orbitofrontal cortex, you could think of as really being the center of flavor processing. So if we take that diagram of smell areas we were just looking at, we can add in some neural pathways that we've seen before, the neural pathway for taste. So we'll put that here below it. Look at the tongue in the bottom left here. From the tongue, remember that taste signals pass through the brainstem. They come out those cranial nerves. They pass through the brainstem, then go through the thalamus. And where do they go? They go to G1, the primary gustatory cortex, which remember, it's in the frontal lobe and also part of that thing I called the insula, the fifth lobe of the cortex nearby. Well, G1 has a lot of connections with the orbitofrontal cortex. It also has some direct connections to like the amygdala and limbic areas, and even some minimal connections direct to the olfactory bulb, but not in a way that generally combines the two senses. Instead, the true combining of inputs, the multisensory processing that combines taste and smell, that happens in the orbitofrontal cortex, which also, by the way, connects to areas we didn't see before, like the hypothalamus and the limbic system and stuff like that. And you don't see it on the picture here, but notice there's some fine print in the top left. Visual signals, meaning coming from the occipital lobe, all the way in the back of the brain, they actually have some projections. Those axons end up projecting neural connections all the way up to the orbitofrontal cortex, as well as other areas like the amygdala. Same thing for touch signals. So somatosensory signals, things like touch, they come in from the parietal lobe. Some of them go to the orbitofrontal cortex, and also to other parts of like the insula nearby and things like that. So the orbitofrontal cortex is really a place that's combining smell and taste and even also like the sight of our food, the touch and texture and temperature of our food in our mouth and so on, along with signals coming, top down from the limbic system and some of the sensory association areas, right? Some of those other areas or arrows going to the orbitofrontal cortex. So as we're already seeing in that last slide, flavor is influenced by more than just smell and taste. So I mentioned, for example, flavor includes somatosensory components, meaning both, texture, but also things like temperature. And indeed, we have identified neurons, individual neurons, that specifically code for like the viscosity of food. Individual neurons that code for fat texture, neurons that individually code for grittiness, for temperature, for capsaicin, like spiciness, for fatty acids. And lots of neurons we find respond to kind of a specific combination of taste plus temperature, taste plus other, some other component. There are others that are unimodal, that respond kind of to a particular taste or separately to a texture or separately to a temperature in an independent way. Unimodal just meaning one modality, one sense, but some also look for that combination. We have a whole bunch of these where you can get this interaction going on between the different senses to help construct this multisensory perception of flavor. By the way, just while we're mentioning like the spiciness, preference for chili peppers, where capsaicin is like the chemical in the peppers that gives them their spice. It's mostly a socially acquired, learned preference, though we do know there's some variance accounted for by the number of papillae on your tongue. Also, by the way, in the mouth, just a side note, in the mouth, capsaicin, so the spice, it desensitizes the pain receptors in the mouth, the nociceptors that we have. So if you're having something really spicy, right, you feel that pain, one trick you can do is just wait for the burn to go away. After that first mouthful, like just suffer through the burn, wait for adaptation after that first mouthful, and then subsequent bites actually don't feel as spicy. We don't end up getting as much nociceptor activation from the same amount of spice subsequently. But anyway, let's get back to some more ways that flavor goes beyond just smell and taste. So let's talk about vision. Visually, the color of something actually affects the flavor we experience. That might seem like, to some people it seems obvious, to others that's pretty damn counterintuitive. It seems like it shouldn't. Like, it might affect your enjoyment of something, right? But why would it affect the actual flavor we experience? Well, a classic study tested this. They showed this very clearly. So they tested people identifying the flavor of fruit drinks, think like Gatorade, basically, when it had the usual colors you would expect to go with a grape flavor, a lime flavor, a cherry flavor, or an orange flavor. Okay, so they're the usual things. Orange flavor had an orange color, lime flavor had green color, cherry was red, and so on. But then in the experimental condition, they hid the color. There was no color. Just give people those exact same drinks, the ones they can usually identify the flavor of, no problem, but give it to them without the visual hint from the color and ask them to identify what flavor it is. And here's the data. This table here has the data from the experiment. The left column here, that's the real flavor of the drink, the actual flavor they were putting in their mouth. And along the top, what we've got is just columns of what the participants reported, like what percentage of people reported a certain flavor when they were tasting that thing. So, for example, if we just look at the top row of numbers here, the top row where it starts with grape on the left, when the drink was really grape, it was actually the grape flavored drink, 70% said it was grape, which is good, right? That's fairly accurate. 15% thought it was lime, which is a little weird. And some had some other guesses. Let's go down to lime. What about when the real flavor was lime? But again, without the usual green coloring, half of the people were able to guess lime, but that's pretty bad. That means half got it wrong. A pretty even split of all the other options of people saying something else entirely sometimes. For cherry flavor, right? If it was actually cherry flavor, but not red, we took away the color, suddenly people start saying they're tasting lime, right? They're more likely to think it's lime than they are to think it's cherry, or they'll guess something else entirely, or even orange. And when the real flavor is orange, we really suck at this. We'll often think it's lime or just something else entirely. And there have been tons of studies like this, by the way. So for another classic example, studies have shown that the more intense color is, we perceive the drink as sweeter. So for like lemon and lime drinks, we experience it as a sweeter lemon, a sweeter lime drink for the exact same amount of sugar in it, just if the greenness or the yellowness is more intense. Likewise, for sugar water that's been dyed red, we experience it as sweeter. If it's more intense visually, we actually experience it as a more intense sweet flavor, or sweet taste in this case. Some more data from the first study actually found something similar. Identical drinks, when they have more food coloring of the appropriate type, are reported as having a stronger flavor. And another study showed it's harder to identify raspberry flavored or orange flavored drinks when they're either uncolored or given the wrong color. So like in the graph here, the black lines above the bars, those are just where we find a statistically significant difference between the bars. And the left half of this diagram, that's for raspberry flavor, right? So for actual raspberry flavor, if it's uncolored or if it's orange or if it's green, we misidentify it about half the time. And for orange flavor, it's not quite as bad. So the right half of the graph, that's orange flavor. The bars on the right. When it's uncolored, you can see though, we're more poor at identifying the flavor. So the visual component affects what we experience, like what we would normally call taste, but what we really mean, it affects our flavor experience. But it's not only somatosensation and vision that get in on this flavor party, even sound can affect flavor. So for example, a pretty classic 2005 study showed that eating potato chips, they'll feel more crisp in the mouth, like that texture component of the flavor. They feel more crisp when the sound is there. If you hide the crunching noise, either hide it completely or just even hide the high frequency components of the sound, you do this by putting it next to a microphone and then you alter the sound before you feed it into the headphones, the noise canceling headphones that they're wearing. If you hide either the sound completely, the crunching noise, or just the high frequency parts of it, then those exact same chips taste stale. People will say this tastes stale and the chips feel softer in the mouth. It's the same chips, and yet we have a very different flavor experience based on the sound. And there have recently been a bunch of studies showing kind of other ways sound affects our flavor experience. Like, just for example, people perceive a bittersweet toffee as being 10% more bitter when they're listening to low-pitched music, but 10% sweeter when they're listening to high-pitched music, things like that. And there are some more, what we might call kind of strictly top-down effects. So not really effects purely coming in from a different sensory component, but from top-down things affecting flavor, like from knowledge, experience, expectation, that more cognitive stuff. Like, the exact same wine is perceived as more pleasant when it has an expensive price tag. But not only that, it activates the orbitofrontal cortex much more, right? The flavor part of the brain actually activates more to the same damn wine just because there was a higher price tag on it. So in this famous 2008 study, the bars at the top here, they're basically a taste test between two glasses of the exact same wine, right? So in the control condition on the left, where there's no price shown, people preferred each glass equally because, duh, it's the same exact wine. But when one of the two glasses of the same wine, when one of the glasses was labeled a $10 wine and the other labeled $90, the expensive glass was rated much more pleasant and the cheap one rated less pleasant than even if there was no price there at all. And in the bottom half of the diagram here, you see the brain activity in the orbitofrontal cortex. We get much more response overall in that $90 condition and actually a depressed response in the $10 condition. And there are other examples like this. Like, here's another example from a different study showing the same basic thing, like the same wine at $5 or $45, or the same wine at $10 versus $50 is experienced very differently. There's other examples of this kind of top-down cognitive effect, like the exact same odor. So you put a chemical smell in someone's nose. The exact same odor is rated as more pleasant when we label it cheddar cheese than when we label that same smell body odor. And it's not just the label here, but like it's not just the smell part, but like the neural response, the orbitofrontal cortex is more active for the cheddar cheese label and less active for the body odor label, right? The thing we wouldn't enjoy as much, suggesting that's kind of the mechanism by which we're ending up with that less pleasant experience when it's been labeled body odor, right? Basically coming through those top-down verbal understandings from the label. So just having that cognitive labeling affects the experience and the neural activity in that flavor zone of the brain. I could go through a lot more. I'll just give a few more examples. So, for example, a frozen strawberry dessert actually tastes about 10% sweeter on average and 15% more flavorful if you put it on a white plate compared to a black plate. Or a latte in a blue mug was rated almost twice as sweet as the exact same latte in a white mug. The shape of a wine glass is known to affect our perception of flavor for the exact same wine. There's studies showing that kids aged like 3 to 5, they report better taste for the exact same food. Like give them the exact same hamburger, the exact same fries. They will report that it tastes better, meaning they get a better flavor experience from it, if it's packaged in a McDonald's bag versus some generic bag that has similar bright coloration, right? Same basic colors, just different patterns, so it's not specifically McDonald's. So what we see is like associations from branding that can also affect our flavor experience, even starting pretty early in life. And finally, but maybe less surprisingly, there's interoceptive input to our flavor experience. Even when we just hone in on like the smell component of eating. So like the pleasantness of a food-related odor, so a smell, like a banana smell or a vanilla smell, the pleasantness of that food-related odor is influenced by your level of satiety. In other words, how full or how hungry you are. So this study, for example, back in 2000, showed that when someone is hungry, a banana smell is equally pleasant with a vanilla smell. So that's the teal bars here, the kind of greenish blue bars here. Then, in this study, they had them eat bananas until satiety, so until they're not hungry anymore. Now, look at the red bars. That's the condition after they've eaten the bananas. Look at the red bars. Both food smells, and again, this is just about the odors that we're testing, but the pleasantness of those odors, both food smells are less pleasant, but the vanilla smell became more like neutral. The banana smell became unpleasant. And when we look at the brain during the same task, specifically zoom in on that orbitofrontal cortex, flavor part of the brain, you can see that the orbitofrontal cortex is basically, it's involved in regulating food intake, our enjoyment, our satiety, because it has all those connections coming in from all these different parts, including top-down experience, interoceptive components, all the other somatosensory components, visual components, sound components, and then of course a bunch directly from taste and smell, and limbic connections and everything else. So, all right, we'll stop there for the chemical senses, taste and smell, and that combined multisensory perceptual experience of flavor.